In these next two videos, we'll talk about DNA repair. What happens when DNA is damaged from external environmental conditions, or when it is when a mistake is made during replication. Mistakes or damage is often referred to as mutations. This can have deleterious effects, causing various diseases. We'll take a look at the amount of DNA damage that occurs each day, and it's pretty staggering. And then in the second video, we'll talk about 3 different ways of repairing 3 different types of DNA lesions. But first, we'll go with some background. So here you have a piece of double-stranded DNA, and a mistake has been made. The original DNA had a G, and there should have been a C over here, but instead an A has been inserted. Now when it comes time for DNA synthesis and these 2 strands separate and both are used as templates for the next generation of cells, this A, if properly replicated, will be a template for T. This G will be a template for the proper insertion of a C nucleotide, which is what it should have been here. And so that 50% in this case of the daughter cells will be mutated at this position, and 50% of the cells will be correct. This is in the absence of any kind of DNA repair. And we'll explore the consequences of this in a few slides. As mentioned, not only can there be mistakes made during synthesis, but there can be lots of damage done to a cell. This is an amazing table which I just saw recently in a talk by Joseph Shiloh, who did some work examining and quantitating the amount of damage done to a single mammalian cell, so to 3 billion base pairs in a single day. And I have to say, I found the numbers to be staggering. I had no idea they would be this large. However, the cells are going to, on average, repair every single one of these mistakes. So our DNA repair machinery is phenomenal in its capabilities. I'd like to demonstrate the consequences of mutation with the first molecularly defined disease, and that is sickle cell disease, sickle cell anemia. It was identified that in 1961 sickle cell occurred because of a single nucleotide change of an A to a T, and this has the consequence of replacing a hydrophilic amino acid, glutamic acid, on the surface of a protein to a hydrophobic amino acid, valine. And we'll discuss in class how this single amino acid mutation causes this sickling effect that you see on the right here of these red blood cells. This is their normal shape. This is the sickled shape. You already have enough information based on the lectures of protein structure to figure that out, but let's discuss that. I also feel compelled to point out that it's been over 50 years since we identified the mutation responsible for the disease, yet we do not have significantly improved treatments for the disease. Your prognosis is not that different if you get diagnosed today with sickle cell disease as compared to when you would get diagnosed back then. There's greater supportive medical care, but not treatments that attack the underlying disease. The point I'm making is that you'll hear all the time in the popular literature, oh, big breakthrough, we found the gene for this disease, but that is still can be an incredibly long way to go before you actually make patients better once you know the disease gene. And you might also ask, why have there been no improvements? And I would say the short answer is developing effective new treatments for disease is really, really hard. One of the ways to avoid having to correct a lot of mistakes or mutations is to not make mistakes in the first place. And the DNA polymerase does this by using several different mechanisms. Here on the top, we see a template strand and a growing strand, and as you can see, an error has been made. Somehow a C was incorporated where there should have been a T base. This is already a rare event, but it does happen. Now the polymerase is coming in and trying to add a second base, but it's going to recognize that the base which it's trying to attach, the new base to the 3' hydroxyl, is not correctly binding the existing template strand, and so that will cause it to recognize not to make an attachment here. And instead, what it can do is it has a 3' to 5' exonuclease activity. That means starting at the 3' end, moving in the 5' direction, it can chew back bases that are not correctly base paired to the template strand. Once the incorrect base is removed, now it can add the proper bases and properly extend the DNA strand. Now this illustrates a very important point, which we will make clearer on the next slide, but it's better thermodynamically to check 3 times pretty carefully than 1 time really carefully. For instance, you could think about how you wanted to spend your energy and time. You could be really, really careful about never putting the incorrect base there. Thermodynamically, that takes a really long time, and it's hard to do when the bases themselves have a lot of similarity. But instead, we'll check 3 times. We won't very often put the wrong one here. That's number 1. Number 2, we won't add the base if it's incorrect. And number 3, we'll excise the incorrect base. So it's, in a sense, 3 attempts to get the proper base present. This slide quantifies how often errors are made during various steps of the replication process and what the overall fidelity is. You can see up at the top that only 1 time in 10 to the 5th base pairs, so is an incorrect base included. Just to be clear, since sometimes people get confused, that is a 10 to the -5th error rate. Then every time an incorrect base gets placed, we'll catch 99 out of 100 using 3' to 5' exonucleolytic proofreading. The times when that is not detected and somehow a 3rd base gets put on, sort of trapping the incorrectly added base, 99 times out of 100, that will be recognized by repair machinery, which we'll see in the next video. So overall, in DNA replication, 1 base out of 10 to the 9th, so 1 base out of 1 billion, is incorrectly included into the genome. Now, you have 3 billion base pairs, 3 times 10 to the 9th base pairs. That means every time a cell divides, you are introducing 3 new mutations into the cell, on average. You might spend some time thinking about how come with all those mutations, we all don't have a great large number of diseases. We'll discuss this a little in class, but it's interesting to think about. In class, we'll also look at some of the quantitative consequences of this trend, or this error rate. Let me give you some numbers to think about. 3e to the 9 base pairs, 1e to the 12 cells, and just for the sake of argument, let's consider a gene that has 1e to the 3 base pairs. I suppose I should have written 3e to the 9 base pairs per genome and base pairs per gene of interest. The results can be pretty scary. So in this slide, we see different locations where DNA can be damaged by the environment. You're not required to know any of these, but as you can see, the base pairs can be affected, the bond connecting the base pairs to the sugar can be affected, the sugar can be affected, or the phosphate group in the backbone can be affected. Damage can be by oxidation, hydrolytic, sorry about that, damage, or by the addition of other molecules, in particular methyl groups. One type of damage that we will talk is one of the most common. It's the one caused by UV light or excessive sun tanning, and that's called a thymine dimer. We can also call it a thymidine dimer. Either is correct, and it's when two T's are next to each other. Normally they have a double bond here, but if two T's are next to each other and it absorbs a photon, you can form a four-membered cyclic ring, and now instead of having two independent bases, you have one larger molecule that's going to take up two spots on the DNA, and your polymerase does not know how to process this when it's replicating the DNA. Fortunately, cells are pretty good at identifying this damage and repairing it. It's interesting to note that this is the exact molecular lesion which causes the body to tan. If you take a cream, and this was discovered at Boston University, if you were to take a cream of thymidine dimers and wipe it on Caucasian skin, that skin will tan just as if it is absorbing sunlight. And it makes wonderful sense. The sun is causing damage to DNA, and it's the damaged DNA that is attempting to defend itself from the sun. Such creams are being developed clinically right now. You are responsible for knowing about this kind of damage, and we'll now break for the next video to talk about the way that cells will repair DNA damage or mutations.